Akshay grew up in India. He studied engineering there. He obtained his bachelor's degree from the Veller Institute of Technology, and then came to UIC as a graduate student in the bioengineering program. But then he transferred to my lab here in biochemistry and molecular genetics, and then transferred to the GEMS program. Since then, he has been studying the role of FOXM1 in liver cancer. You will see that he has done a lot of work in this area. He has published 2 papers as co-authors, as second author, as actually a really significant second author, and then submitted, in the process of submitting a co-first author paper. Last year, he presented his work in the AASLD meeting, where he received the AASLD President's Award for his presentation. So today he's going to talk to you about the role of FOXM1 in RAS-induced hepatocellular carcinoma.
All right. Thank you, Pandey, for such a nice introduction and not making me nervous. So, and finally I'm here. Thank you all for coming to listen to my defense talk. One of the proteins of interest from our lab is FOXM1, and the organ of interest for our lab is liver. And I got interested to find out what is the role of FOXM1 in RAS-induced hepatocellular carcinoma. So to begin with, I'll give you a brief introduction about liver cancer. It's one of the most common types of cancers found prevalent around the world. By the end of this year, the estimated number of new cases that are going to happen in the world is going to rise to 700,000, whereas the death toll because of liver cancer is going to rise to 600,000. Now it is intriguing because these numbers have sort of tripled since the 1980s, and that is because of rise in the hepatitis infections, as well as increase in the incidence of non-alcoholic fatty liver diseases. HCC, or hepatocellular carcinoma, is the most common type of liver cancer and contributes to about 3/4 of the cases. I wanted to see how the expression of FOXM1 relates with high-grade progression of HCC. So we took some help from Dr. Guzman and quickly found out that FOXM1's overexpression correlates with the high-grade progression of HCC. Over here in the slide, I present tumor sections from tissue sections from the tumor biopsies of HCC patients. Grade 1 tumors are comprised of well-differentiated tumor cells, whereas grade 3 have a very high accumulation of poorly differentiated tumor cells. As it is clear that grade 1 tumors, which are primarily comprised of well-differentiated tumor cells, express little FOXM1, whereas when we look at the grade 3, there is a very robust expression of FOXM1. The stagings of FOXM1 has been quantified in the box plots below and shows a very significant change when we go from grade 1 to grade 3. Before we get into the story, I would like to introduce FOXM1 as a transcription factor briefly. FOXM1 is a transcription factor that belongs to FOCAT family of proteins. And it is recognized by the presence of a conserved winged domain that FOXM1 uses to bind the promoters of its DNA gene targets to regulate their expression. FOXM1 is known as a pro-proliferation factor, and that is because all the dividing cells need to express FOXM1. The cells that are not dividing or are terminally differentiated express very little or undetectable levels of FOXM1. FOXM1, as we can see from the schematic over here, has a very pivotal role to play in the cell cycle. FOXM1 activates the expression of STIP2, which leads to proteolysis of P27, activates the expression of CDC25A, both of which are needed for the G1S transition. FOXM1 transcriptionally activates the expression of CDC25B, crucial for the SG2 transition. It activates the expression of chloride kinase, or RABI, which are important for the mitotic progression and centrosome maturation. Apart from this, FOXM1 provides protection to the cancer cells against oxidative stress. In a transformed cell, the oncogenic stimuli leads to the production of reactive oxygen species, and their rising concentrations lead to oxidative stress. In this process of increasing ROS concentrations, the increasing ROS triggers the expression of FOXM1, which in turn activates expression of certain ROS scavenging genes such as catalase and aminosulide, which keep the rising ROS concentrations under check. Thus, FOXM1 provides protection to the cancer cells against oxidative stress, as well as helps them to proliferate. Now, coming to a few regulators of FOXM1. Oncogenes and growth factors in general are known to activate the expression of FOXM1, whereas tumor suppressor proteins have been shown to inhibit its activity. For example, ARF over here has been shown to directly bind to FOXM1, translocate to the nucleolus, and render it transcriptionally inactive. We spoke about cell proliferation and oxidative stress regulation by FOXM1 in the previous slides. In the context of cancer, it has been vividly studied that increased FOXM1 expression leads to increased tumorigenicity. FOXM1 has been shown to activate the expression of certain EMT markers and has been shown to increase epithelial to mesenchymal transition. Increased FOXM1 leads to increased cell migration, as well as its expression helps in establishing a pre-metastatic niche so that these circulating tumor cells can colonize the secondary site. FOXM1 has also been shown to confer drug resistance to certain DNA-damaging agents. Since the process of EMT cell migration and establishment of a pre-metastatic niche are all very crucial to metastasis, we often also refer to FOXM1 as a master regulator of metastasis. There's another function of FOXM1 that was discovered by a previous grad student from our lab, Janai. What she found that FOXM1 inhibits the process of differentiation, and she showed this using the mouse mammary gland. What she showed was FOXM1 transcriptionally inhibits the expression of GATA3, which is the master regulator of mammary luminal differentiation. We'll come back to that later. So now, since I was going to work with FOXM1 and cancer, liver cancer, I thought of doing some literature survey and found that the RAS signaling pathway is ubiquitously active in HCCs. But when we look at it, we see that the active RAS mutations contribute to only very low percentages of HCC cases. How RAS signaling pathway is ubiquitously active is because the RAS regulatory proteins, GAPs, which have the responsibility of switching off RAS by switching them from a GTP bound state to a GTP bound state, are silenced. And that is because their promoters have been hypermethylated. We see over here that the RAS gap proteins, RASL1 and DAP2IP, are silenced in about 3/4 of the SCC cases. Not just that, the upstream activators of the RAS gap proteins, for example, in this case, PITX1, which is known to activate the expression of RASL1, is silenced in about 40% of HCC cases and more than 80% of HCCs with poor prognosis. So to study the role of FOXM1 in liver cancer, in RAS-induced HCC, I utilized a previously described transgenic mouse model. In this mouse model, the mutant RAS gene, HRAS12V, is expressed under the albumin promoter. As a result of this, there is constitutively active RAS signaling in the liver of these mice, which leads to several hepatic alterations of these in these mice as they age. This has been summarized in the table that I'm going to show you next. So this is the summary of all the hepatic alterations that these mice go through as they age. However, I would like you to focus in this part. Over here, the mice are 9 months old, and about 90% of male mice show incidence of hepatocellular carcinoma. This incidence of hepatocellular carcinoma is less than 30% in the female mice, and this is quite reflective of how we observe HCC incidences in human patients. decided to use this model to find out the role of FOXM1. We crossed this mouse with an already available strain in the lab. This strain allows us to conditionally delete FOXM1 by the activation of MX3. So the resultant triple transgenic mouse, we aged these mice to 9 months and then induced the expression of the CRE by addition of a double-stranded RNA molecule or PIPC. We activated the CRE, deleted FOXM1 using 5 doses of the double-stranded RNA molecule. So let's see what happens. Over here I'm showing you the expression of the transgene in these transgenic mice, non-tumor tissue as well as tumor tissue. When we look at the non-tumor tissue, there is a positive expression of the transgene, indicating that the RAS transgene is properly expressed in these livers. However, when we look at the tumor tissues, the HRAS V12 mRNA shows much higher expression. When we look at the expression of FOXM1 mRNA from the non-tumor tissues of the transgenic mice, they show a very comparable expression to the FOXM1 expression from a wild-type liver. But when we look at the tumor tissues, they show a much higher expression of FOXM1. Next, I'm going to show you what happens to the expression of FOXM1 and RAS when we induce the CRE and delete FOXM1. On the left panel, I'm showing you the FOXM1 mRNA before and after deletion. There are 3 tumor tissues from 3 different mice before deletion. And these are 3 tumor tissues from 3 different mice where we induced the CRE and deleted FOXM1. We clearly see that there is a decrease in the FOXM1 mRNA expression. However, there is still some remnant amount of FOXM1 that shows up in our qPCRs. When we look at the expression of the transgene, there is practically no effect of FOXM1 deletion on the expression of the HRAS12 mRNA. We took the tumor tissues from FOXM1 deleted and undeleted mice, and we stained it for FOXM1. On the left, we have a tumor section from a mouse where we did not delete FOXM1. And we see that at least 2 types of cells, one with a round nuclei and one with an elongated nuclei, they show positive expression of FOXM1. When we induce the CRE and check for FOXM1 deletion, we see that only the cells which have the round nuclei, or which are the HCC cells, lose the expression of FOXM1, whereas the other cells, the ones with the elongated nuclei, still retain the expression of FOXM1. And this is where this remnant mRNA expression might be coming from. So what happens to the liver tumors when we delete FOXM1? We compared a group of 21 mice where we did not delete FOXM1 to a group of 25 mice where we induced the CRE and deleted FOXM1. In the top panel, there are 4 representative livers from the group of mice where we did not delete FOXM1, and we see multiple tumor nodules on the liver. Upon deletion, the number of these tumor nodules decreases. This decrease has been quantified here in the box plot and shows a very significant change upon FOXM1 deletion. However, we were still seeing incidence of tumor on some livers, like the tumors were small in size, less in number, but the tumors were still there. And so we wondered if a prolonged activation of the CRE or a better efficient deletion of FOXM1 would help us to improve our results. So instead of using 5 doses of the PIPC, we induced the CRE with 10 injections of PIPC. And our results definitely showed improvement. We compared a group of 6 mice where we did not delete FOXM1 to a group of 7 mice where we used the CRE and deleted FOXM1. The number of tumor nodules, the differences in the number of tumor nodules further enhanced, as we can see over here. Also, we also found 2 livers that were completely tumor-free upon FOXM1 deletion. This decrease has been quantified over here and shows a very significant change. So what happens at the molecular level when we delete FOXM1? We checked for proliferation of these tumor cells upon FOXM1 deletion. we stained the tumor sections from FOXM1 undeleted mice and deleted mice for PCNA, which is a marker for proliferation. We see that these brown positive nuclei indicate the presence of PCNA in the tumor sections where we did not delete FOXM1. These PCNA positive nuclei show a drastic reduction upon FOXM1 deletion. The decrease has been quantified over here and shows a significant change. Next, we looked at some of the proliferation genes, which are also target of FOXM1. We looked at CDC25B, Aurora B, and poro-like kinase 1. I had introduced them earlier in my presentation. These are FOXM1 targets and show a decrease in the expression when we delete FOXM1. This was true for 2 sets of the tumors that we analyzed for QRT-PCR. We looked for changes in cell death. We isolated fresh tumors from the mice and made single cell suspensions, stained them with an FITC tagged antibody that detects active caspase. Active caspase indicates early apoptosis. And so over here we see that almost negligible amount of cells show active caspase 3 staining, whereas the cells where FOXM1 was deleted show enhanced apoptosis. This change has been quantified over here and shows a very significant increase in the number of apoptotic cells upon FOXM1 deletion. We reconfirmed this finding using tunnel assay. Tunnel assay is used to detect double-stranded breaks and is also a hallmark of early apoptosis. We see that in the tumor sections where we deleted FOXM1, there are higher number of tunnel-positive nuclei indicating higher apoptosis in the tumor cells. Next, we wanted to look at what happens to the concentration of ROS in these tumor cells. So we stained frozen tumor sections from FOXM1 undeleted and FOXM1 deleted mice, and stained them with DCF-TF fluorescence to check the presence of ROS in the cells. To see it better, I'll just switch off the slide. Clearly, we see an increase in the fluorescent cells upon FOXM1 deletion, indicating there is a much higher accumulation of ROS in the tumor cells upon FOXM1 deletion. This has been quantified over here and shows a very significant change. So I'll summarize this part so that we can move ahead. Till now, I hope that I've convinced you that FOXM1 deletion leads to inhibition of RAS-induced liver tumors, as well as it does so by leading to lower proliferation, increasing cell death, and it leads to higher accumulation of ROS in the tumor cells. A few years back, in 2013, Michael Karen's group identified the presence of liver cancer progenitor cells. What they found was that the liver cancer progenitor cells are the cells that are able to initiate liver tumors, and they found it positive for 2 cell surface markers, CD44 and EpCAM. We got interested to find out if FOXM1 deletion leads to the changes in the number of these cells. And so we stained the tumor sections from FOXM1 deleted and undeleted mice and found that the membrane staining of CD44 that we see in the FOXM1 undeleted tumor section is drastically lost upon FOXM1 deletion, indicating that FOXM1 deletion leads to drastic loss of the CD44 positive cells, HCC cells. We next looked for EpCAM positive HCC cells, and we saw a drastic loss of EpCAM positive cells as indicated by loss of membrane staining upon FOXM1 deletion. This decrease in the number of cells, EpCAM positive cells, has been quantified over here and shows a significant decrease in the number upon FOXM1 deletion. We confirmed this finding using Western blot. We checked for the expression of CD44 and EpCAM from FOXM1 deleted tumors and undeleted tumors. We see there is positive expression of CD44 in the 4 tumor samples where we did not delete FOXM1, and we see quite a robust expression of EpCAM, which is lost upon FOXM1 deletion, as seen in the right half of the Western blot. We wanted to see if FOXM1 deletion leads to any changes in the stemness genes. And we found that upon deletion of FOXM1, there's a decrease in the expression of the PMI analog and C8 mRNAs. And this was true for 2 sets of the tumors that we analyzed with qPCR. And a short summary for this part. FOXM1 deletion leads to drastic loss of CD44 and NETCAM positive HCC cells, as well as FOXM1 deletion leads to changes in the expression of stemness genes, decrease in the expression of stemness genes. So till now I've told you that in a normal liver, when we have constitutively activated RAS signaling, it leads to HCC with high FOXM1 expression. And then when we target FOXM1, it leads to a disproportionate loss of CD44 positive cells, NETCAM positive cells, leads to higher accumulation of reactive oxygen species in the HCC cells, which leads to inhibition of HCC progression. So, since expression of FOXM1 correlates with accumulation of poorly differentiated cells, since FOXM1 is important for the survival of the CD44 and NETCAM positive cells, as well as FOXM1 is important for the expression of stemness genes, we wondered if FOXM1 regulates the expression of liver differentiation genes. And to test that, I was interested in FOXA1 and A2 because these liver differentiation genes have been vividly studied in liver development as well as liver cancer. And they have significant differentiation roles to play. So I again stained the tumors from the mice where we had not deleted FOXM1 and deleted FOXM1. Clearly, we see increase in the positive brown nuclei which are stained with FOXA1 upon FOXM1 deletion. This increase in the number of FOXA positive nuclei has been quantified over here and shows a significant increase upon FOXM1 deletion. We reconfirm this finding by assaying for total protein lysates from the tumor tissues, and the 5 tumor tissues where we deleted FOXA1 show an increased expression of FOXA1 protein. We also found this to be true for FOXA2. Over here, we hardly see any positive FOXA2 positive nuclei, but on the section on the right, when we have deleted FOXA1 from the tumors, we see a robust increase in the expression of FOXA2's positive cells. This has been quantified here on the right and shows a very significant change. We reconfirm this finding by assaying for FOXA2 protein in the total protein lysates from these tumor tissues, and we see a clear increase in the expression of FOXA2 protein in the FOXA1 deleted tumors. So next thing that we wanted to see is whether the same pattern is followed in the human HCC patients. So for that, we again took some help from Dr. Guzman and obtained tissue microarrays from human HCC patients. I had already shown you that FOXM1 expression is highest in the grade 3 tumors, whereas there's a very little expression of FOXM1 in grade 1 tumors. FOXA1 and A2 show opposing patterns though. Grade 1 tumors that have a better prognosis and are comprised of well-differentiated cells show a very high expression of FOXA1 as well as A2. But when we look at grade 3, there's practically very little expression of FOXA1 and A2 that I found in the tumor sections. These changes have been quantified in the box plot on the right, and we can see that expression of FOXA1 and A1 and A2 show opposing trends. Now, at this point, we thought that FOXM1 is kind of negatively regulating the expression of FOXA1 and A2. And so to confirm that, we utilized some in vitro techniques. We took human HCC cell lines, HU7, and overexpressed FOXM1. When we overexpress FOXM1 in these cell lines, we see a drop in the mRNA expression, a significant drop in the mRNA expression of FOXA1 and A2. The Western blot from the cells shows the same. when we attenuate the expression of FOXM1 using siRNA, there's an increase in the expression of FOXA1 and A2 mRNA. The Western blot suggests the same. When we knock down FOXM1, there is a significant increase in the protein levels of FOXA1 and A2. Now at this point, we were almost certain that we are seeing a negative regulation by FOXM1 on FOXA1 and A2. And this was very interesting because a previous grad student, Janai, had studied this. What she found that FOXM1 recruits retinoblastoma and DNA methyltransferase 3B to the promoter of GATA3, hypermethylates it, and renders it transcriptionally inactive. Now, luminal progenitor cells in the mammary gland, they require GATA3 to mature. When GATA3 levels fall down, the luminal progenitor cells cannot become mature luminal cells, and so there's a higher accumulation of these progenitor cells. So to test if the FOXM1 regulation on FOXA1 and A2 was a direct one, we carried out immunoprecipitations. Over here I'm showing you that FOXM1 binds to one of the two putative sites that are present on FOXA1 promoter. This is 3091 base pairs upstream of FOXA1 transcription site. And this is around 4000 base pairs upstream of FOXA1 transcription site. We see that DNMT3B as well as RB, they show a very robust binding at the same place where FOXM1 is bound to FOXA1 promoter. But when we attenuate the expression of FOXM1 using siRNA, we see that DNMT as well as RB fail to bind on those sites, indicating that FOXM1 is required by these proteins to bind onto those sites. And we like to say that FOXM1 recruits DNMT3B and RB to FOXA1 promoter. In this immunoblot, this is a co-IP experiment wherein I show you that FOXM1 binds to RB and FOXM1 binds to DNMT3B in these cell lines, in the cell line that is being used, which is WHO7. We tested the occupancy of FOXM1 on FOXA2 promoter. FOXM1 was bound to 2 of the sites out of the 4 putative binding sites. DNMT3B and RB, they showed a robust binding at those particular sites as well. When we attenuate the expression of FOXM1 using siRNA, we lose the binding of DNMT3B as well as RB at the same sites, indicating that FOXM1 is recruiting DNMT3B and RB on the FOXA2 promoters as well. Since DNMT3B is involved in this complex, we wondered if there are changes in methylation on the promoter that happen. So we found the presence of multiple CpG islands on both FOXA1 and A2 promoters. And a lot of them showed changes in methylation, but over here I'm just presenting few sites that were very close to FOXM1 binding sites on FOXA1 and A2 promoters. We see that when we overexpress FOXM1, there is increase in the methylation of the promoter on both FOXA1 and the 2 sites on FOXA2 promoters. When we knock down FOXM1, there's a loss of methylation on the same sites, on the same CpG islands. This is true also for in vivo cases. I isolated the genomic DNA from FOXM1 deleted and undeleted mice, did a bisulfite conversion, and checked for the changes in methylation at the mouse CpG islands, which are kind of different from the human CpG islands in the position. And we see that there's a loss in the methylation at promoters, on the CpG islands of mouse promoters, upon FOXM1 deletion. And this was true for both FOXA2 as well as 2 CpG islands that I found on FOXA1 promoter, indicating that loss of FOXM1 leads to loss of methylation at FOXA1 and A2 promoters. So what is the role of RB in this complex? To investigate that, we re-engineered the WHO7 cells to inducibly knock down the expression of RB by addition of doxycycline. Let me take you through this graph a little slow because it's a little busy graph. And we tested the same islands that I've shown you in the last slides. In the first one, we just express control vector in presence of RB. In the second one, we overexpress FOXM1 in presence of RB, and we see increase in methylation. We knock down RB, but do not overexpress FOXM1 over here. In the yellow bars, we overexpress FOXM1 in absence of RB, and we clearly see that FOXM1 is unable to induce any changes in methylation levels in absence of RB. And this was true for all the three CpG atoms which were found on FOXA1 and FOXA2 promoters. So what is the biological significance of this mechanism? To find biological significance, we carried out 3 in vitro experiments: clonogenicity assay, soft agar assay, and ability of FG2 cells to form spheres. When we overexpress FOXM1 alone, it leads to a huge increase in the number of colonies that are formed. This has been quantified over here and shows a very significant change. But when we co-express either FOXA1 or A2, the effects of FOXM1 are kind of negated, and we see a drop in the number of colonies, indicating that FOXA1 and A2 are somehow negating the effects that were carried out by FOXM1. These have been quantified over here, and we see a significant drop in the number of colonies when we co-express FOXA1 or A2 with FOXM1 as compared to expressing FOXM1 alone. Same was the case with soft agar assay. Soft agar assay is used to test the anchorage independence of the cells, which is a hallmark for cancer cells. When we overexpress FOXM1, it leads to increase in the number of colonies, soft agar colonies. When we co-express either FOXA1 or A2 with FOXM1, the effects of FOXM1 are negated, and we see a drop. And we see a drastic drop in the number of colonies. Next, we looked at sphere formation assay. Sphere formation assay reflects stemness properties of the cancer cells. When we overexpress FOXM1 alone, it leads to bigger spheres and more number of spheres. When we express FOXA1, A2, or A1 alone, it leads to very low number of cells, very low number of spheres, and these spheres are very distorted. I won't even call them spheres. When we co-express A1 or A2 in combination with FOXM1, we see that the number of spheres that are formed are almost as close to the control vector expression samples, indicating that FOXA1 and A2 negate the effects of FOXM1. So we wondered if FOXA1 and A2 also regulate the expression of FOXM1 in a feedback loop. So to test this, we expressed control vector in the first link, FOXA1 or FOXA2 alone in WHO7 cells, or we co-expressed FOXM1 with A1 or FOXM1 with A2 in WHO7 cells. Now, when we look at our FOXM1 plasmid is flag tagged, so we can check the expression of the exogenous FOXM1 by Western blot against flag. We see that the exogenous FOXM1 is equally expressed irrespective of whether FOXM1 plasmid was expressed alone or in combination with FOXA1 or A2. But when we look at the total FOXM1, which is up here, we see that FOXM1 shows a very drastic blasting signal actually when it is expressed alone. But if it is expressed in combination with either FOXA1 or A2, the total FOXM1 levels are not that high. So what is happening over here? To understand that, I would point to a study that was published by Water Lab a while back. What they found is that FOXM1 is able to induce its own expression in a positive autoregulatory loop. And what we thought is that when we co-express FOXA1 and A2 with FOXM1, somehow FOXA1 and A2 are inhibiting this autoregulatory loop. To find this out, we overexpressed FOXA1 and A2 in the WHO7 cells and checked for the mRNA levels of FOXM1. When we overexpressed FOXA1 and A2 in WHO7 cells, we saw a significant drop in the FOXM1 mRNA expression, indicating that this positive feedback loop of FOXM1 is somehow perturbed by the presence of FOXA1 and A2. We also saw a loss of FOXM1 target genes such as survivin, Aurora B, and CD44 upon FOXM1 or A2 overexpression. And then we wondered if FOXA1 and A2 are sort of binding over here and directly inhibiting the this autoregulatory loop. So we carried out a chip experiment again, and we found that FOXA1 and A2 are actually bound to FOXM1 promoter very close to the transcription start site, which is only about 600 base pair upstream of the transcription start site of FOXM1. So I know that the story is kind of getting interesting at this point, but this is all the data that I have to show you over here today. And we are still working on it to figure out the feedback loop. So to summarize this part, I would say, I hope that I've convinced you that FOXM1 inhibits the expression of FOXA1 and A2 in human and mouse HCCs. And that FOXA1 and A2 oppose the effects induced by FOXM1, and that is possibly because they interfere with FOXM1's expression. This is the graphical summary of my work. FOXM1 binds to FOXA1 or A2 promoters, recruits DNP3B and RB, hypermutates it, renders it transcriptionally inactive, and that's how your well-differentiated tumor cells, which have a better prognosis, they actually turn to poorly differentiated tumor cells in absence of FOXA1 and A2. And what we think is that this is a war between the FOXM1 and FOXA factors, which will decide the fate of the liver tumor. If FOXA1 and A2 win the game, they somehow inhibit the effects of FOXM1 by a mechanism that we are still trying to dissect. And that will lead to well-differentiated tumor cells and a better prognosis of HCC patients. In case FOXM1 wins the game, that shall lead to accumulation of poorly differentiated tumor cells and leading to a higher grade progression of HCC. So with that, we'll come to the most important slide today, acknowledgement. So starting with Pradeep, obviously. Back home in India, gray hair is a sign of wisdom. And modern medicine, modern research has also told us that it can be a sign of stress, right? So it was like a year and a half back that Pradeep and I noticed that each other's hair are getting grayer faster. And so we decided it was time for me to graduate. I'm still not sure whether it was my blossoming wisdom or just his pity for my stress, which made me stand over here. But jokes apart, it's been a very fulfilling experience working in Pradeep's lab. And I think I've grown as a scientist and a human being because of the time that I spent there. I have not memorized this, so I'm just taking notes over here. Please bear with me. So, Pradeep carried out a routine with me, which I don't think is very different from growing bacteria in the flask. He would inoculate an idea into my head, obviously being the empty flask, and then let it grow for a while. He would check for the growth by engaging me into conversation in the corridor, and after the right amount of time, when he thought the log phase was reached, he would allow me to plan and carry out the experiments. He always made sure to carry out stimulating conversations, which were scientifically very stimulating, irrespective of the topic of research, irrespective of whether the topic was related to our active research in the lab or not. However, a lot of conversations that I treasured happened at the lab lunches and were quite outside the scope of the lab work. these conversations were about life experiences, and they ranged from consciousness to neuroscience, from Greece and Socrates to Krishna and Karma, to politics, Donald Trump, and science. And so I actually felt sorry for myself when I missed one of those lab lunches because I was on a holiday in India. He's seen me throughout the ups and downs of the grad school and has always supported me, as well as provided a very conducive environment where I could balance my personal and work life. So thank you, Julie, for that. Okay, so he's one person that I can probably describe in one word as a guru. So, Pradeep, thank you very much for being my greatest support. All right. I would like to thank Angela, Karen, Nissim, Vadim, and Deepak for their suggestions throughout this course of my grad school. Their suggestions have improved the quality of work that went into my thesis, and they have always given me thoughts to chew upon during the committee meetings. The best thing about this committee that I really liked is that everyone was very approachable. Whenever I had a problem, be it a personal problem, technical problem, or a problem with a fundamental concept, I would just walk into their office and get the help right there, just like that. So thank you very much for being so supportive. I would like to thank Dr. Guzman. She's a very patient person. She waited for almost a month before I could stain the slide correctly. Whenever I went to her, she would never say that you wasted the slide. She would say that, okay, maybe you need to change some antibody concentration and things will be fine. And in this process, she used to also tell me about the pathology of the tissues. And so these interactions have now made me capable enough of deciding whether a particular tissue is a tumor or not, and whether, if it is a tumor, what grade it is. So thank you, Dr. Guzman, for that. Now, coming to lab members. This is going to be interesting. When I arrived in Pradeep's lab, Dragna had just started her postdoc. And since, as Pradeep mentioned, I was from engineering background, he thought I needed much training in the basic sciences, and he thought it was a good idea that I work with Dragna. And man, was that tough. So as engineers from India, we are used to doing things at the last moment. With Dragna, it was parallel processing. 4 experiments running in the lab, and then we had to go to the mouse room to take care of the cages. So, and it was crazy for me. Well, this apart, we are very good friends. And I think she's one of the smartest people that I've met. She can think like a mathematician, physicist, and biologist, and gives the best suggestions and explanations by connecting the dots. Vaibhav joined our lab in 2014, and we planned a new project of studying FOXM1's repressor function in context of liver cancer. We planned the experiments together, did troubleshooting together, and Vaibhav has contributed significantly to the work presented today. In fact, both Dragna and Vaibhav, without their presence, the present project wouldn't have been rendered as I showed you. So thank you guys. I would like to thank Dr. Nishit Shua for their contribution to this project. Dr. Nishit worked on the dissection of FOXM1 activation and repressor function. Shua always asked us very intriguing questions about our project, which led to a new perspective and new dimensions for the progress of the project. And thank you, Jim, for ordering the stuff on time so that our experiments never got delayed. And all of these people that I mentioned have helped create a very friendly environment in the lab. So thank you all. I would like to extend my thanks to the previous lab members, or PRADs, as some of us refer to. Especially Nilupal Roy, Janai Kar, Zebin Wang, Dr. Damiano Pantiri, Megan and Colleen. The department labs also merit a very big thank you for maintaining a scientific stimulating environment in the department and for sharing reagents, and also for teaching me stuff that our lab did not actively do. It's been wonderful meeting so many students from so many different countries and so many different cultures over here in this program. And you guys are some of the smartest people that are there. And I have a sense of pride associated that I'm going to graduate from the same program that you guys are part of. So thank you guys. And you guys have always created a very scientific and fun environment, so thank you for that. I would like to extend my thanks to the GTSs, Dr. Caffey, Dr. Catherine, especially Dr. Catherine, because she was very patient with me when I forgot to send my self-evaluation, student self-evaluation forms for 3 months. She would just send a kind reminder with just one exclamation mark. Please respond. And I would do that then. So thank you, Dr. Catherine. A special big thank you to the CCTS PECS scholarship for funding me for a couple of semesters. And at last, I would like to thank my family and friends, just like in all award speeches. You guys, this is my big chance of an Oscar-like speech, and I'm not going to give up on it. My family was wonderful. I have to say though, that I threw some of their plans. Despite both my parents holding a doctorate degree in their own fields of management and education, I kind of blew them away when I told them that when I graduate from this program, I won't be young anymore in the Indian context. And so it kind of blew them away, and also their plans of getting me finding me a nice girl to settle down with. So yeah, my sisters were very supportive all the way as well. And they are from very different fields. One of my sisters is from humanities, the other one is an urban planner. And they always tried to sound very excited when I told them what I did in the lab. And one of them also got to the point where we had an ethical debate about the use of transgenic animals in the lab. So thank you for that. And my friends have been very kind to me throughout my stay in Chicago as well as the United States. And have always tried to make me feel special and happy, and always kept me entertained. So a very big thank you to you guys, and we will party tonight. And with that, hopefully I think I've covered everyone. And I'll take any questions. Thank you. Yes, Dr. Kat.
When you introduced your mouse model, which you were going to use, you made a point of the fact that there's a much higher propensity rate of tumorigenesis in the male mice than the female mice,
Yes.
which is also true in humans.
Yes.
But then, unless I missed it, you didn't really get back to that. So did you use only one gender?
Yeah, we only utilized male mice because the incidence of the tumors is almost with 90% penetrance over there. So we found more tumors to analyze. And there are hormonal differences in how these liver tumor numbers are governed in male and female bodies. So we probably would have found the same things in the female mice. Yes?
So one of the big inducers of hepatocellular carcinoma is hepatitis C virus, as I understand it. BNC, I guess, but mostly C.
Mostly it's C. It contributes
C.
alone to 50% of the HCC cases.
Yeah,
Yes.
so is there something specific about hepatitis C as a reducer of FOXM1 or an inhibitor of FOXA1 and A2 that plays a role in human liver cancer?
That part I won't be able to answer, but what I do know is that HCV viruses, they require a high constitutively active RAS signaling to replicate. So maybe they lead to overexpression of the RAS signaling, and that's how they lead to tumor. But the mechanistic studies have not actually elucidated this very vividly yet. Yes, Shuwei.
FOXA family has 3 members.
Can you speak a little louder, please?
FOXA family has 3 members.
Right.
A1, A2, A3.
Yes.
They are highly homologous.
Yes.
They bind to the same DNA sequence. And if you study FOXA1 and A2, what happens to A3?
So we've seen the regulation of FOXA3 as well. It's just that I didn't have enough time to show you the results of FOXA3. We see that FOXA3 is also negatively regulated by FOXM1 in the same cells that I showed you, except the story about FOXA3 wasn't totally complete, so I did not present over here. Yes?
So you are using the MX3 to delete, right?
Right.
You're not concerned that this is actually not deleting only in the hepatocyte, but also in the hematopoietic cells and probably other cells, either in the liver and also or outside the liver.
Right. So the idea of this project was to kind of provide a similar scenario of what a drug might do. So if there is a drug that affects FOXM1 levels, it's also going to do that in the blood cells.
I understand, but do you think that the effects that you get
Yes.
are not only due to the effect on the hepatocyte, but rather other cells as well?
We have not seen that. We did not check that. That's a good point, and we are actually working on projects which are going to elucidate that. Yes, Dr. Katz.
So in this presentation, FOXM1 seems like the bad guy, right? It promotes tumor genesis,
Yes.
basically. So is it a good guy too? Is it an essential gene?
Oh yes, it's one of those genes that have to be balanced properly. If you don't have it, then it leads to embryonic lethality and it leads to a lot of problems in the organ development, such as liver. But if you have too much of it, it'll lead to accumulation of poorly differentiated cells by the mechanism I showed, and that will lead to aggressive phenotype of the tumors.
Is FOXM1 a target for drug development? In this regard, is anybody focused on that?
I think people have tried to target FOXM1. Dr. Gartle's lab has come up with thiostreptin. Is that it? Yeah, thiostreptin as a molecule to target FOXM1. Technological developments have to happen a little more before we can start like specifically. The targeting has to get better with the technology. I think that needs a little technological development. Yes.
So I noticed that the U7 cells do not express or express very little of FOXM1.
U7 cells?
Yes.
They actually express quite a bit of FOXM1.
But when you did the experiment, I didn't see much. When you overexpress, you see it. So why did you
I mean,
have
that's
to,
just an exposure of the Western blot. I can go
so
back.
why did you have to overexpress it?
Because I mean, I'll show you the effects.
No, but I understand. Okay, they have some.
I mean, because
why do you have to overexpress?
we
in order to
overexpressed
see
as well
it?
as knocked down the expression of FOXM1, and we saw the same regulation in both the cases. When we overexpressed FOXM1, the effect was a little slightly lower at the protein level, probably because FOXM1 levels are already very high in U7 cells. But when we knock down, the FOXA1 and A2 proteins show much enhanced expression, and that could be a reason why we see.
Okay.
So do you have the same result with the electric mice?
I'm sorry?
Do you have the same result with electric mice?
We have not tested that yet. We have not. Yeah.
So you've shown very interesting interactions between FOXM1 and the FOXA factors.
Right.
Are there similar interactions with any of the other FOX family of proteins? Because I think FOXO3 is increased with reactive oxygen species, and that has its own consequences in autophagy and so on. So are there other interactions amongst this family when FOXM1 is going up and down?
You mentioned FOXO, and FOXO and FOXM1 have been actually shown to compete against each other for the VEGF promoter, and I think it is a very context dependent how they'll behave in the context of what kind of cells that you're dealing with, and yeah. But yes, there have been interactions shown with the other FOX family proteins. It's just not this one.
Yeah, it's actually a very good point because FOXAs and FOXO, they actually evidence that they cooperate in DNA binding, promoter binding. So I I think there's things to it that we have not been able to study so far.
Yes, go on.
So your RB, the FOXM1 binding site on RB, where is that compared to E2F? Like say, can they compete?
Good question. So, I have done this experiment when I was in second year, long time back. So, and I did see that when you overexpress FOXM1, the E2F that is bound to RB is reduced by 50%. I saw that in MCF7 cells, in 450, MDMB 453 cells. But yeah, I did not pursue that. So I think there is definitely competition between E2F and FOXM1 for RB binding.
So there are 3 FOXM1 isoforms.
Right.
Are you talking about FOXM1B?
I'm talking about FOXM1B.
Right.
As this isoform is overexpressed in liver cancer.
Have you looked at the other isoforms?
I have not done that.
And sometimes with your antibody, you see a smear. Are those different isoforms?
No, I would think that those are different phosphorylated forms.
Is your antibody specific for 1B?
No, but our plasmids and siRNA are.
All right, if there's no other questions, guys, remember there's a research, if it passes, there's a research.
Right, thank you all for coming.